DNA structure and replication, and RNA structure. Table of contents: genetic transformation, discovery of DNA, DNA structure, DNA replication, RNA structure. Genetic transformation. DNA. The precipitate that you see in the bottom of the flask here is deoxyribose nucleic acid, DNA. DNA is the informational basis for all living things. It stores instructions to build living things. Every molecule, organelle, cell, organ system, organism, and population is built according to the code in this molecule. It is the building block of the genes that control how bodies are shaped and how organisms react to environmental factors. The rather innocuous-looking precipitate, DNA, provides the information for the shape and function of the complex basilisk lizard and all life on Earth. The selfish gene. Individuals are not stable things. They are fleeting. Chromosomes, too, are shuffled into oblivion like hands of cards soon after they are dealt. But the cards themselves survive the shuffling. The cards are the genes. They merely change partners and march on. They are the replicators, and we are the survival machines. When we have served our purpose, we are cast aside, but genes are denizens of geological time. Genes are forever. From Richard Dawkins, evolutionary biologist and Oxford University professor. In other words, genes live beyond individuals. As an example, blue eyes are a phenotype, a physical trait controlled by a single gene. A recent study showed that a mutation in one individual's OCA2 gene, which produces the pigment that gives color to eyes, created a gene for blue eyes. This occurred 8,000 years ago, and the new gene was passed generation to generation. Today, approximately 560 million people have blue eyes. Each individual carries 2 copies of the original mutation. The gene has long outlived the human in which it originated. Primary discovery. Nucleic acids were first isolated by the Swiss physician Friedrich Miescher, who in 1869 discovered a microscopic substance in the pus of discarded surgical bandages. At the time, it was unknown cellular substance and was not considered important until many years later. The search for the genetic material. The molecule that was responsible for inherited traits had not been discovered in 1928, when Frederick Griffith was trying to make a vaccine for pneumonia. He used 2 different strains of the bacterium Streptococcus pneumoniae, S and R. The S strain of the bacterium kills mice. The R strain does not. He was surprised to observe the following during his experiments. When S strain bacteria were killed with excessive heat and then injected into mice, the mice survived. However, when a dead S strain was mixed with a live R strain and injected into mice, the combination was lethal. Griffith concluded that the dead mice had live S strain bacteria in their blood. The S strain bacteria could reproduce. His observation led to scientific questions. What is the molecule that is responsible for virulence? Why can it be transferred from dead bacteria to living bacteria? He called this observed phenomenon transformation. Griffith investigated why the virulence of the S strain was restored when it was combined with the R strain. It is easy to tell the difference between R and S strains because they look different when growing on a petri dish. S has a smooth edge, as you see here, and R has rough edges, as you see in the image here. So here are the observations that led to these further scientific questions. The S strain bacteria kills mice. The R strain bacteria does not. If you kill the S strain by heating it, it does not kill mice. But if I take this killed S strain and combine it with live R strain and then inject the mice, that kills the mice. And a dead mouse, the blood from the dead mouse contains living S strain. So something happened here where this killed S strain, when mixed with live R strain, resulted in living S strain. Here's Griffith's conclusion. The living R strain took up a chemical left from the dead S strain. This chemical transformed the living R strain into the deadly S strain. More investigations were required to identify the chemical responsible for the transformation. We now know that the chemical taken in and incorporated into the living bacteria is DNA. In this case, the gene that produces the deadly toxin transformed the living R strain bacteria into lethal S strain bacteria.